Well, good afternoon, everybody, and thank you so much for your patience, and thank you so much for attending our September webinar, which is very highly anticipated because it's going to be on something that's very topical. If you don't know about this, where have you been? It's on COVID, and the title is Leveraging Single-Cell Genomics to Understand COVID-19. And this seminar talk will be provided by Dr. Alex K. Shalek, who's a co-member of the Institute for Medical Engineering and Science, and an associate professor of chemistry, as well as extramural member of the Koch Institute for the Integrative Cancer Research at MIT. So this will be for about 40 to 50 minutes of a presentation. And at the end, we'll have time for question and answer a discussion as well. So my name is Pablo Chauke, and I'm part of a team of organizers that make sure that we have monthly webinars. I'm not gonna read the whole abstract because I'm looking at the time, but we have, please Google him. Dr. Alex has an illustrious career, has won many awards, works at prestigious institutions, and I think some of you would want to collaborate with him. And without further ado, Dr. Alex, thank you so much for allowing and agreeing to give this presentation. Go ahead and give your presentation.
Oh, it's my absolute pleasure. And thank you all so much for having me. And thanks for accommodating me by 10 minutes. I had to drop my son off at school here in, right outside of Boston and run back across the street. So if it feels like I'm catching my breath at the minute, it is because I am catching my breath, but I'm delighted to be here with you all. So thank you so much for the opportunity to speak. You know, what I really wanted to do today was to tell you a little bit about some of the work that my lab has been doing in collaboration with a number of other individuals over the last several months now, you know, almost 2 years, it feels like, trying to understand what's happening in COVID-19 and contribute in any way we possibly can. And I'll walk you through some of the science we've been doing, some of the ways in which we've been using existing data sets, some of the new data sets we've been creating. But I hope that this sparks a conversation around some computational methods, around some new experimental technologies, and would love to hear how we might partner further to bring some of these technologies to bear to COVID, but also to a number of other problems that are of relevance to your community. So, as was introduced, I don't think I have to explain that COVID is a global pandemic. Usually I would be very excited right now when I'm giving the seminar because I would be with you and I would be looking forward to a visit. But obviously I'm doing this from my home because of this ongoing pandemic that has led to, hundreds of millions of cases, millions of deaths, and, for which there's actually been tremendous responses if you think about how rapidly vaccines have been developed and deployed. I think that if we think back to the beginning of COVID, even though it feels like it was so long ago, when we started, really sort of around the early part of 2020, there was sort of this idea that there was a new condition that was arising that had these characteristics of, a respiratory disease. When you start talking about what we were seeing symptomatically at the level of the individual, there were conditions that were commonly associated with the upper respiratory tract, like cough and shortness of breath. In a subset of patients, there were gastrointestinal issues, and there's just this general inflammation or malaise that associated with COVID-19. And so, from the early indications and the early descriptions, we got a good idea of what was happening at sort of the high level. And by some beautiful work that was built off some work that had been done previously, thinking about SARS-CoV-1, a number of groups figured out what was the virus, SARS-CoV-2 or coronavirus. They figured out what was the receptor that it used, ACE2, and the accessory proteases like PMDRSS2, that enabled it to latch onto cells, infect them, release its RNA, and then begin replicating and creating new virus. In seeing these two things, one of the biggest problems is that it's hard to connect them together. Much of this work was done in culture models where people were using cell lines and working with viruses. Much of this was happening in humans. And early in the pandemic, we began to wonder, what cells in the human host are actually being targeted by the virus and to what effect? How can these things that are happening at sort of a subcellular level relate back to what we're seeing at the level of the individual? Now, in many places, asking questions like this would require generating new data sets, going out and collecting samples from patients, which is obviously hard in an ongoing pandemic, particularly when you don't really know what's going on and you don't know the safety constraints associated with it. But one of the reasons that we and a number of other individuals thought that we might actually be able to contribute here is because we had generated these huge repositories of single-cell RNA sequencing data And as I'll show you in a second, these data, even though they were collected for a different purpose, gave us an opportunity to start to ask questions of relevance to SARS-CoV-2. And this is one of the things that I like most about genomics, is that because you measure everything, you can very often go back and look at existing data sets to ask questions that you previously hadn't anticipated. And so, as much as any given experiment that generates a data set is important, I think in a lot of places, the data is equally important because it lets you go and do new discovery-based efforts. And so here on the left, I show you a non-human primate, a rhesus, and a number of different tissues labeled by color. And on the right, I'm showing you a plot of sort of about 100,000 single cells where each line connects cells based on how similar they are. They're colored by tissue, and each one's a point. And I'm going to tell you how this is generated really quickly so it seems less abstract, but for now you can think of it as a beautiful picture. So let me talk a little bit about generating single cell sequencing data and why we thought it might be useful in the context of understanding COVID-19. So over the past several years, there have been transformative advances in molecular and cellular biology and in nanofabrication and micro devices that have enabled us to take tissues that we used to profile, break them down into their individual cellular components, and then isolate those cells. And so these could be things like Eppendorf tube that you might be familiar with, where you put one cell in one of these 1.7 milliliter tubes. It could be a multi-well plate. It could be something more complicated, like an integrated fluid tip, something made out of microfluidics, or something that uses droplets, or something that uses wells, or so on and so forth. And by basically reducing down the container size, we're able to isolate individual cells, and then by adding specialized molecular reagents, tag information from them. And so here I'm showing you an example where we use picowells, where basically we isolate each individual cell in its own little room. This little box, it's about 50 microns by 50 microns by 50 microns. With it, we put a little bead that can interrogate all of the messenger RNA that's on the cell when it's lysed. So now what we can do is within this well, we can lyse a cell, separating this cell from its neighboring cell from its neighboring cell. We can take advantage of the fact that all of the messenger RNA, everything that the cell is thinking about creating with respect to protein at a given moment is polyadenylated. Use an oligo DT primer to bind to it, reverse transcribe, PCR, and amplify up so that we get many, many copies of that cell, enough to actually profile. And then we can go and use next generation sequencing to look at what the cell contained. And while this may sound silly or funny or even impossible, there's really been this transformative advance where instead of doing this on a bulk population, we can actually now do this on a single cell. And so you can not only do this on one cell, but by using these methods, you can parallelize and do it on tens of thousands or hundreds of thousands or even millions of cells at once. And so what that means is that for those 10,000 or those 100,000 cells that we might be looking at, we can look at the expression of roughly 30,000 genes at once, see what they're thinking, get billions of data points, and then do some computational analyses to try and figure out what this data might be telling us. So you can imagine that right now we're thinking about this collection of cells as a huge data matrix that's 100,000 by 30,000. What we can do is then we can go and try and figure out what the structure of this data is. And a way that we do this is we try and remove data points that aren't good. We try and figure out what are the most variable genes across these things so that we're really focusing on what's different, because the similarities won't tell us anything about what separates cells. We do some simple dimensionality reduction, followed by some different methods for figuring out which cell is similar to which cell, almost like creating a social network. And then we do some clustering to basically figure out groups. And so here I'm going to do a movie where in real time we're doing this through iterations of computation, and I'm labeling things by the color of the tissue from which the cell derives. And cells that are more similar are going to be next to one another, and ones that are more dissimilar will be pushed apart. And so when we run this video, what we end up with is sort of this hairball that eventually starts to self-organize. You can see that as I run iterations, cells that are similar get pulled together. Things that come from common tissues end up in similar spots. Other things separate out, and you can begin to see that we're starting to get what looks like some organized structure. Here, the lines are a little thick. So what we can do is we can now simplify this by thinning those lines down. And what we have is this hairball that I was showing you before, where each of these small little points is now an individual cell, and the connections between them tell you something about the similarity of them. So really, you can think of each of these points as representing, a 30,000 dimensional vector, where basically we know the expression of all 30,000 genes for each point. And then, we can start to begin to identify what these different groupings are. So we can see some groups are defined by tissue. These ones right here are gray. And when we look at the genes that enrich them, we can see things that help us to figure out that they're microglia and therefore shifted to that tissue. We can see things like oligodendrocytes. In other places, we see things that are refined to the liver, like hepatocytes. In some places, we see populations like eosinophils that actually spread across multiple different tissues. And interestingly, many of the populations that are in the middle here that are shared across many different tissues are immune cells. So, you might ask yourself, well, this is great. What does this actually have to do with COVID-19? Well, because we have this incredible array of data where for each individual cell, we know what the expression of each individual gene is, we can now begin to ask ourselves across tissues, what are the cells that express ACE2, this entry receptor for SARS-CoV-2, and what express the accessory protease that helps to get into cells TMPRSS2. And so by looking at the data that I just showed you from a non-human primate, what we find is that there is tissue-specific expression of this receptor that lines up with what we'd expect from the symptomology. So we don't see a lot of expression in the bone marrow, maybe a little in the CNS, relating to symptoms there, a lot in the ileum and the jejunum related to the GI symptoms that I mentioned previously, expression in the lung, and not necessarily in some of the other tissues in the body. But what we can do is instead of just asking about, what tissue this is expressed in, we can look at one specific tissue and dive in and ask what are the cells that are actually likely to be infected. And so here I'm showing you some data that was generated in collaboration with some partners at RE in Durban, South Africa. We're looking at human lung tissue, and each individual point is now going to be a cell. I've done the same sort of clustering and identification of cell types based upon genes that I showed you previously. So you can see from the lung, we capture some T cells, some mast cells, some fibroblasts, some pneumocytes, some macrophages, and so on. If we go now and look at these individual cells, and we look at the expression of the genes that we care about, here ACE2 and TMPRSS2, we actually see something surprising. And that's that ACE2 really only appears to be expressed in these ciliated cells and in these type 2 pneumocytes. TMPRSS2 is a little bit more broadly expressed, but shows up in those same populations. And so, when we started looking across tissues, what we found was actually something surprising, which is that, there were defined cell types and targets that made sense. In the nasal epithelium, we saw secretory goblet cells and ciliated epithelial cells that expressed both ACE2 and TMPRSS2. In the lung, the type 2 pneumocytes and the epithelial cells, and in the gut, absorptive enterocytes. But when we looked at the fraction of cells that were ACE2 positive or ACE2 TMPRSS2 positive, you can see that actually it's incredibly small. And that really didn't make sense, when we were thinking about sort of needles in a haystack, given sort of the symptoms that we were seeing and how fast this was progressing across the population. So you might say, well, this is great. You're looking at existing data, but how do you figure out what cells are actually infected and to what end? And so, this was a question that we started to ask ourselves. There were beautiful studies that were coming out in the literature that were starting to figure out what was happening in ex vivo cultures. Some staining in vitro where you could see now where SARS-CoV-2 was, but you couldn't necessarily resolve what was the actual cellular target. It was hard to figure out which ones were virus positive. On the other hand, we are seeing that there were some interesting systems level effects of infection that had to do with broad inflammation, increases in things like the expression of IL-6, changes in the expression of interferon stimulated genes where there was less ISG expression in those who had more critical disease in the periphery. Major differences compared to what we were traditionally seeing in the context of tissues within the blood. So, this left us with sort of open questions as to, what's really happening in vivo and, what's happening in target tissues where you can actually, first see infection. And so, we got sort of inspired by the fact that if you look at the technologies I was showing you, we could work with increasingly small amounts of material. We began to wonder, instead of trying to collect large tissues, which are prohibitive, could we in some places take advantage of readily accessible tissues that we could sample to ask some of these questions? And so I'm sure many of you have unfortunately had the opportunity to have a nasopharyngeal swab or an NP swab to test for coronavirus. And so normally you would take one of these NP swabs, you put it in a tube and somebody would run PCR to figure out whether or not you had any viral nucleic acids and tested positive or if you did not. Well, what we began to wonder is, could we use these swabs for more? So what I mean by that is when you think about what the swab does, it actually goes into your nose, it sloughs off a little bit of the cells that are on the surface, just like maybe the experiment you did in your high school biology where you took a toothpick and scraped your cheek and looked at it under a microscope. We wondered, would there be enough cells here that we could take these tubes, isolate what came out, and then perform single cell RNA sequencing on them to figure out what cells are there and which cells are harboring virus. So to make a long story short, we spent a lot of time developing a pipeline that would let us do this, where we take nasal pharyngeal swabs, freeze them very rapidly, and then figure out how to extract cells from them using a series of different steps, which you can now see on this protocol that's available online, and then isolate that in individual cells for sequencing. You might say, well, do you get viable cells out of these nasal swabs? And the answer is yes. If we go back and we do immunohistochemistry on some of the things on the slide, you can see the ciliated cells right here with the cilia. Actually, they swim when we look at them under a microscope unless you fix them. We can see goblet cells that are MUC5AC positive. We see squamous cells. So, we thought that this might actually be a place where we could begin to say, well, what are the first targets of infection and to what effect? So with partners down at University of Mississippi Medical Center, which has unfortunately been hit incredibly hard by COVID, as well as partners here in Boston at Boston Children's, we began to collect a cohort of individuals where we were looking at nasal swabs to figure out what might be happening in COVID. So we collected a number of swabs from SARS-CoV-2 negative individuals, a number of healthy matched controls. We had some non-COVID-19 ICU individuals that had severe respiratory ailments and were put on respirators, but not due to COVID, as well as a number of individuals who went on to have mild or moderate COVID, and some that went on to have severe or critical COVID. And we sort of separated this based upon who's scoring, where 5 and below was mild to moderate, and 6 and above was severe. And so, we can look a little bit more thoroughly at some of the demographics of what we have here. And, we have a good breakdown of the demographic based upon what sort of the surrounding populations are, good balance of males and females, and, a good number of age ranges, highlighting that even though, in certain places there's increased susceptibility as you become older, this is something that actually affects everybody. So across these 58 samples, we're able to recover about 32,000 single cells. And here on the right, I'm showing you, one of these graphs of the single cells that we recovered, where each point is an individual cell. And I'll walk you through this really quickly so you can see what we were getting out of the nose. We have a number of different epithelial populations, these basal stem-like cells that are up here running through some, developing cells that are differentiating to some, functional effector cells like goblet cells, and some secretory cells moving through another intermediate to these ciliated cells that help to sweep some of the stuff out of your nose. Over here, we have a number of squamous cells. We also collect a number of immune cells that come in, inclusive of T cells, B cells, dendritic cells, macrophages, mast cells, and even some more, nuanced, recently described cells like ionocyte and enteroendocrine cells. And, because this is sort of surface scraping up in the nose, we don't collect everything that you might imagine. We get a pretty good representation of, what's happening on sort of the surface layer of the nasal mucosa. Now, what we can do is we can take the same map right here and we can ask ourselves, well, what changes in individuals that are positive for SARS-CoV-2? And what we see is that actually there's this dramatic difference in which cells we're detecting in individuals who are negative for virus by PCR versus those that are positive. You see that a lot of what's changing is really in some of these populations right here that have to do with epithelial subsets. So to really dive in a little bit more, what we did is we actually subset this data. So as I told you originally, when we generate these maps, we look at variable genes across all of our populations. But what makes a secretory cell different from a ciliated cell might be washed out by what makes a secretory or developing ciliated cell different than a macrophage, because these epithelial cells might be much more similar than some of these immune populations. So what I've done now is I've gone and I've taken out everything that is not an epithelial cell and I've recreated this map, clustering again. And here, we can see sort of the same trajectories that I showed you previously. We have our basal cells down here, which are stem-like cells. We have, some of our intermediate progenitors here, which move into some of our more terminally differentiated cells like ciliated cells or squamous cells. And to figure out, how these cells relate to one another, we can take advantage of a computational technique called RNA velocity. And so, what we're doing when we do single-cell RNA sequencing is we're taking a snapshot, but, not all of our cells are synchronized. We have cells that are in different states, moving from, maybe basal to differentiating a little bit more. And we can try and infer where cells are migrating using some of the properties of the data itself. So in RNA velocity, what we do is we look at our mRNA, and by looking at the ratio of unspliced to spliced RNA, we try and figure out what transcripts were more recently produced, i.e. those ones that have higher intron retention. And what this does is it gives us an idea of where a cell is going as opposed to where it's been, which is dominated by the mRNA that's already mature. So by doing this, using this package called scVelo, we can overlay on top of our data sort of an arrow telling us about where our cells are moving. And what we see, not surprisingly, is that from these stem-like cells, we appear to be migrating up into these more terminally differentiated cells that come across the secretory intermediates, which then seem to be moving across to these cells over here that are developing ciliated and ciliated with squamous on its own island. And this lines up pretty well with what we think happens based upon the literature, where we have basal cells that go to the suprabasal cells, which then either go to goblet or secretory cells, which might be able to interconvert. And so you can see we've gotten up here. And then we move through these deuterosomal intermediates to these developing ciliated cells and mature ciliated cells. So we have this map. Now we can begin to ask ourselves, well, what is changing in SARS-CoV-2 infection? Well, when we start to look at it, the first thing that we see when we add virus is that actually there's a tremendous change in the number of secretory cells we have. So if we look, there's a major increase in the fraction of the population that are secretory epithelial cells. Not surprising, you're trying to clear virus, maybe something's trying to happen here, but there is this major shift where you're getting this diversification increase. You might say, is this just by sequencing? Is it an artifact of what you're looking at? No, we can go and we can stain using antibodies and sort and show that there is an expansion of secretory cells in individuals that have more severe disease. The second thing that we see is that there's really a loss of some of these more mature differentiated ciliated cells. And so if we look here, what we see is that we move from healthy down through these non-ICU individuals to these moderate to severe. And what we're seeing is a dramatic reduction in the fraction of these sort of terminally differentiated cells. Now, with an expansion here and a differentiation here, you might be asking yourself, are you moving back through some of the intermediates? And the answer is, it appears to be yes. It looks like we're sort of getting an expansion of some of these precursor cells, these deuterosomal cells, as we get to more severe disease. So it looks like a de-differentiation of some of these most important populations right here, and an increase in some of these more intermediate, less terminally differentiated populations. So, you might say to yourself, well, that's what's happening globally. But what's happening within individual cells? This is sort of the shift in the overall abundance. But if we were to just control, is a mature ciliated cell in an individual that has SARS-CoV-2 infection identical to one in an individual that does not? And so when we started looking at this, what we found was something that was actually really surprising. So if we go and we look at control individuals, ciliated cells from them, versus individuals that have COVID-19 that just hit mild or moderate, what we see is that there are a number of genes that are significantly upregulated in the individuals with COVID. If you start to look at these genes, maybe you see many of your favorites. I know I do, IFIT1, MX1, some of the HLAs, IFIT-I, IFIT-M1, MX2, all things that we canonically think about as being associated with an interferon response. And so we see that when we do some enrichments here at the bottom, based upon average fold change, that there's a significant increase in the interferon response in COVID. If we go look at our control versus our more severe COVID individuals, similar things here. HLAs, you can find some of the genes that are in here, but as I start to search, what you recognize is that many of these interferon response genes that were popping up before aren't coming through so clearly. And this gave us a first hint that became more clear when we started comparing what was happening in mild to severe disease. When we did, what we saw is that many of these genes that you think of as being canonical, being essential for antiviral response, like IFIT-M1, STAT1, IFI44, and so on, were actually much more strongly upregulated in the individuals that had mild disease than those that had more severe disease. And this wasn't just something that turned out to be true when we looked at ciliated cells. It turned out to be true when we looked at all of our epithelial populations. So if we looked at some of our goblet cell subsets, like a subset that was MUC5AC high, or the SCGB1A1 high population, where here I'm defining it by some RNAs that they express very highly, you see that there's a strong upregulation of interferon response in mild to moderate, but a major decrease as we go down to those who have severe disease. Same thing in secretory cells, same thing in deuterosomal cells, and even in ciliated cells. And so what it looks like is in individuals who have severe disease, there's a major blunting of this antiviral interferon response early. And you might say, what else could you sort of tell me about this? Are you seeing that that this holds across donor? And the answer is yes. If we do this by cohort and we look, we see that there are much stronger responses when we look broadly across the people that have mild to moderate disease versus those who have more severe disease. And this doesn't have to do with exactly when you sample or the timing of when we collect, because if we look, individuals who we collected when they were only mild to moderate but went on to have severe disease actually had decreased responses to virus through interferon early. So what I'm really telling you is that actually what it looks like is that an early lack of high interferon response seems to be associated with progression towards severe disease. So you might ask yourself, well, if interferon is so critical, who is producing it within the system? And so we can go back now and we can start to ask ourselves, across all of our populations, which cells are expressing various interferon stimulated genes or other cytokines or chemokines that we think about. When we look, we see that actually we're detecting very, very little interferon across all of our different populations. There's maybe a little bit of interferon gamma coming up in some of our T cell subsets, but we're really not detecting any of the interferon alphas or beta or gamma in any of our subsets here. And for the aficionados in the group, you might say, well, I know a number of these technologies have a hard time collecting interferon. Is it possible that what's happening is that actually, your technology just can't capture it because of the structure of the transcript? And I don't think that's the case. When we look at some swabs that we took, here, these are nasal washings actually in the context of influenza. What we can see is that we do get good expression of interferon alpha and good expression of interferon beta. So it's not that we can't detect these transcripts, using the technology at all. It just really looks like we don't get it in COVID-19. And to give us sort of an indication that this makes sense. Actually, there are a number of other studies that have shown now that one of the things that's defining about SARS-CoV-2 is the lack of interferon response that comes with it. And so if we compare, for example, what happens in a mock infection to influenza, where you see a strong induction of interferon beta or interferon lambda, you can see that in SARS-CoV-2, there's very little induction. Same thing shows up in the periphery, where you look at plasma and you see very little interferon lambda when you look at COVID-19 relative to healthy, whereas in influenza, you see an incredible increase. And sort of what we're seeing here in these target tissues of infection are very similar to what people were seeing in the periphery, where they would see in neutrophils, for example, in a study out of UCSF by Max Krummel's lab, that there was a major decrease in the amount of interferon response among individuals that had more severe disease. And other things where people had shown that host genetics played a role. And we're saying that exactly the same thing is happening here in the target tissue. So it's not there's just a decreased peripheral response, but it actually looks like that there's a decreased response at the site of infection in this sort of intrinsic antiviral response that seems to be associated with disease severity. Now you might ask, why do some individuals respond less strongly than others? Well, some people that started to show genetic links started to talk about autoantibodies against interferon. And so, about 1 in 10 people, if you've been following the literature, have been shown to develop antibodies against interferons, which you could imagine would be driving severe disease. And so, in asymptomatic individuals or in healthy individuals, you really don't see antibodies against, various interferon subsets. And in our cohort, when we look, we do see evidence of autoantibodies, but we only see them at the same percentage as the rest of the population. So we don't see them in our healthies or our mild moderates. In our severe, we find one person where they had autoantibodies. So this may explain a little bit of what's happening, but it doesn't explain why we're seeing these major differences in interferon response. You might ask, well, is it a difference in sort of host genetics? And there have been these nice studies from Jean-Laurent Casanova showing that, defects in specific genes related to interferon response can lead to, increased severity of disease. And we don't think this is likely to explain it based upon sort of the percentage of individuals that had these DNA variants, but we don't have genotyping here. So one of the biggest questions we set out to ask in all of this was not just what was changing, what was the impact of infection, but more broadly, what are the targets of infection? Which cells actually harbor virus, and what is the virus doing within those cells? Because that may give you an idea of how you might prevent infection, or what are the pathways that you could block or potentiate in order to reduce viral infection. So we remember from sort of the thing that I showed you at the beginning in the non-human primates, the putative targets of the virus in the nasal epithelium were sort of these secretory goblet cells and ciliated epithelial cells. But now that we have actual nasal epithelium from infected individuals, you might say which cells have virus, and are there things that are specific to cells that are infected versus ones that are bystanders to the infection? And so how are we going to go about doing this? Well, I told you that the strategy that we use to capture messenger RNA is we take advantage of these polyadenylated tails on the messenger RNAs. It turns out that SARS-CoV-2 also has a polyadenylated genome. And so the same strategy that we're using to capture messenger RNA can be used to capture the transcripts of the virus itself. So when we look across our cohort, we can see that the number of transcripts or unique molecular identifiers that we capture aligning to SARS-CoV-2 in healthy individuals is 0. We get 1 errant read in 1 individual, none in the non-COVID ICU, a couple spurious reads in the convalescent. And then we get major increases in the mild and moderate case and in the more severe case. And about 3% of all the cells that we get in the nasal swabs appear to have virus within them. Now, one of the issues when you're looking at this is that some of the viral reads that we could have could be due to bona fide infection or could be due to ambient infection. And so they could be due to other cases, and we want to convince ourselves that what we're measuring is real. So the first thing that we did in sort of looking at this is we sort of correlated what we were detecting in terms of the number of viral reads against qPCR. And what we saw was this beautiful correlation telling us that we were measuring something that was quantitative as opposed to just semi-qualitative. Second, we spent a lot of time correcting for ambient contamination to remove viral transcripts that were coming from viruses that were in solution as opposed to being inside of cells. And this was done using a ambient correction technology. I'm happy to talk through this. But basically what it does is it looks at everything that sits in the soup that doesn't get collected from cells and uses that to subtract out from your cells what it doesn't think are bona fide transcripts. And there's some cool technology there that we could talk about if you want to think about sort of computational methods that are being applied and developed here. But even more than that, to give ourselves confidence, we could take advantage of the fact that it's not just a transcript, but it has this interesting life cycle that's associated with its replication. So we know that SARS-CoV-2 is a single positive strand of RNA, and like a number of other coronaviruses, it replicates by synthesizing a complementary full-length negative strand. So it comes in, you get this negative strand, and then above and beyond that, it slices itself into a number of the pieces that it needs. So it creates these subgenomic regions that encode for structural proteins. Then it goes through and it splices together different sequences, so that it takes a leader and it puts it onto these different elements here, removing these transcription regulatory sequences to create these subgenomic RNAs. And so what we can do is we can actually look not just at reads to virus, but we can look at reads to strand based upon sense, and also to some of these various truncations that exist. And from that, we can figure out which cells we think are really bona fide in their infection, which ones actually really harbor virus. And what we find when we do this is that we find virus as being associated with some of our subsets like goblet cells, these KRT13 high cells that we call HeLiC-like, some squamous cells, some developing ciliated cells, and some ciliated cells that really express high ISG. And so these are places where we have high viral UMIs post ambient correction. We have coverage of the viral genome. We detect negative strands and we detect subgenomic alignments. And I'm happy to dive into this more if anybody wants. And you might say, well, now that you've actually looked at this, how useful was some of the data that you've collected originally? Does it actually line up with what you would have expected? And so what we can do is we can make a plot of this. And what we find is if we look at the original data that we generated back on our paper in 2020, looking at the fraction of ACE-positive cells versus the numbers that we detected now, that actually there's this really nice correlation. So if we look, we predicted that there'd be relatively high infection of goblet cells based upon their expression of ACE2, and that's what we see here. You might say, well, what's off diagonal? Where did you not do so well? And the place where we didn't do so well are in some of the macrophage populations. But when we look at sort of the structure of the virus within them, it really looks like these cells are doing what you expect a macrophage to do. They're eating infected cells, they're gobbling up virus. So, we actually think this is driven by sort of the function of the cells as opposed to being bona fide infection. All right. So as I'm showing you here, not all cells in an individual population are actually positive for virus. So you might ask yourself, well, what is different in the cells that are infected? How does the virus change some of the gene expression that exists within these populations? And could that tell you something that you might target therapeutically? And so, we could ask, is there an intrinsic response? Is there something that makes the cell more suitable? And so if we look at our data, what we can do is we can find genes that within a specific population are higher. So here, for example, in a subset of goblet cells that are consistently higher in viral positive cells versus those that are consistently higher in bystander cells. When we look over here on what's sort of upregulated in cells that actually express virus, there are a lot of things not surprising. You can see that, there's higher interferon responses in cells that are infected. And if we do sort of a overall enrichment, what we see is there's antiviral defense, antigen presentation, cholesterol biosynthesis, and some of the spike proteases that you would think are important for infecting cells. On the other side, if we look at what's higher in bystanders, you see the ability to sense interferon, interferon receptor alpha 1, antigen presentation via MHC2, and some serum amyloid proteins. And what this suggests are sort of things that you could try and go against if you wanted to, protect yourself against infection in the cell or potentiate it. And now you might ask yourself, well, you told me the amount of interferon response is important, and I'm seeing there's more interferon response in these cells that are infected. Does this difference that you're talking about in sort of intrinsic immunity still hold true when we start to think about cells that actually have virus? Well, what we can do is we can actually go subset on, all cells that are viral positive now. And we can say, do we see a difference in the number of virally positive cells between mild and moderate? And we don't. We don't see a difference in the amount of positive strand virus that we detect or the amount of negative strand virus that we detect. But what we do see is a major difference in these infected cells in the fraction of cells that are expressing these interferon response genes. So IFN-M3 is down, MX1 is down, STAC2 is down, and so on and so forth. And you might say, well, is it just that there's less response in severe, or is there something that defines severe? And what we see is that actually what's really upregulated in the individuals that have severe infection are some of these inflammatory genes, such as S100A9 or calprotectin, among others. So what I really had hoped to show you was sort of what we're showing here was that there's a data set that we've generated that's now publicly available that you can go look at and where you can explore data. You just have to go down to this link here at the bottom. I forgot to update this. It's now published in Cell and it's open access, so you can go look at it. What we were able to do by taking these nasal swabs is we could basically identify the full epithelial trajectory. So what cells exist, how they differentiated. We found that there was some major rewiring of the epithelium in COVID, with increases in goblet, deuterosomal, and secretory cells, and a loss of some of these more terminally differentiated cells like ciliated cells. We established pipelines that would help us to figure out which cells were infected, what stage of infection they were at, and this is something that here we're doing for SARS-CoV-2, but it's equally applicable if you want to study something such as HIV or Ebola or whatever infection you might be interested in. And we've done work in both of those I'd be happy to talk about. And really what we find that's sort of surprising is that there seems to be this defect in intrinsic immunity in sort of interferon response by epithelial cells that's associated very early with the course that you're going to take. And so a question that you might have is, could you actually potentiate this response? Could you try and make the epithelium more responsive as a way of protecting not only against SARS-CoV-2, but against the next respiratory pathogen? And I think this idea of sort of the role of non-conventional immune cells in helping to mount immune response is going to become increasingly important. We're going to have to think about how we potentiate these things within tissues for future instances. So, this is all in the nose. You might say, well, what does the nose, sort of this first site of infection, do relative to everything else? Well, to ask questions about what was happening, in sort of the lung and in other tissues. We partnered up with a number of individuals around the Boston area to do what we call the, Boston COVID-19 autopsy study. And this was an incredibly complicated study for a number of reasons, as you might imagine. Sort of at the start of the pandemic, everything was being done in BSL-free facilities. All of us were trying to minimize who was in the lab, for safety considerations, and trying to figure out how we could contribute, but also do it in a way that people were comfortable and where we weren't putting people at risk. And so by taking a few individuals from a number of labs across the road, Mass General Hospital, Brigham Women's, the Beth Israel, MIT, and the Ragon, we partnered to go look at what was happening in multiple different tissues that were infected or that were affected by disease. So thinking about lung, heart, kidney, liver, so on and so forth, trying to understand what was happening in disease tissue relative to a healthy reference, and then trying to figure out what was happening in the cells that were infected, much like we just did in the nasal swabs. And so we performed 17 autopsies with our partners at the hospital, collected a number of different tissues from participants, and got a number of different tissue specimens with replicates. And I won't go into everything here. I'm happy to answer questions, but I can sort of give you a flavor for what we saw. We took lung tissue, sort of right downstream of the nasal epithelium, sort of the next site where, we get a lot of the scarlet disease. We did something very similar to what I showed you previously. We did some stuff to correct for ambient RNA because you have a lot of dead and dying cells. And so there's free floating RNA. We did a lot of QC to remove things that were problematic, did some clustering to find our groups. And we did two different things here. One was we tried to manually annotate. We went through and we looked at each individual population, tried to figure out what each cell was based upon genes. We also tried to build some, machine learning based approaches that would help us to classify cells rapidly. And this becomes important when we have a new data set and we want to interpret it in the context of lots of other data sets. So traditionally, the way you do this is you bring together all the data sets, you cluster them, and then you try and say, well, this is how this lined up with the previous data set. But what we wanted to do here was to sort of transfer labels from previous data sets to these new data sets to annotate them quickly. And so, what I really want to highlight in just sort of a nutshell is that we see very similar things in the lung. So if we look at what changes between diseased tissue and healthy reference, we see a major change in the relative frequency of some of these more terminally differentiated epithelial cells. Here in the lung, we're talking about AT1 and AT2 cells. But we see that in COVID-19, there's a substantial reduction. And there's an increase in sort of these intermediates in individuals, and also in sort of these pathological responses that are associated with wound healing within this target tissue. So sort of what we get in all of this is that the infection is basically remodeling the epithelia and driving to an intermediate state, probably through some dedifferentiation, some transdifferentiation, as a way of trying to deal with some of the stress response. You might ask yourself, you're seeing similar things in sort of composition. Are you seeing similar things with respect to viral targets? And what we find when we look at the lung is that, similar cells are the ones that are actually being infected. We detect some epithelial populations where we get some viral reads. We also, get a lot of cells that appear to be eating infected cells like myeloid cells. And one of the big problems with working with the lung, particularly later, is that many of these target cells of the epithelia that you would normally think would be infected are actually now dead and gone. And so we don't detect them very well because they don't have a lot of viable RNA to look at. And we see, once again, major inductions in inflammation among our myeloid populations, sort of linked to what you might have associated with severe disease. So, I think that what I wanted to highlight from all of this is that, what we're really trying to do here is we're trying to figure out what was happening in sort of the human biology of all of this, as a basis for understanding what's next. I think that, one of the things that our lab and many others have been trying to do a lot more as of late is, rather than starting with models, to start with, complex human diseases with partners around the world to sort of figure out what's happening, in infections in target tissues, so that we can then go select models that are appropriate to recapitulate important features of the disease. If I'm studying tuberculosis, it may be great to have a non-human primate, but it might not be appropriate to have a cell line or a mouse depending upon something that I might want to study. And so by thinking about what we're looking for first, and then figuring out the right system in which to test our hypotheses, I think we can get to a place where we can accelerate interventions back to humans much more rapidly. And so in the context of COVID, we did exactly the same thing. Once we were figuring out what was happening in humans, we were trying to figure out what models were appropriate for studying some of the aspects of disease. And so in parallel, in studies that I don't have time to talk about today, we've been working on looking at, what happens in primary challenge in the context of non-human primates. And so looking longitudinally at pedobronchial or alveolar lavages from non-human primates, sort of longitudinally, where we see similar things, early responses among some macrophages with distinct responses that happen later, and then some interesting inflammatory responses later. And this is in monkeys, and these animals go on to have very mild courses and resolve. Whereas on the other hand, if we look at another model like the Syrian golden hamster, it does a better job of recapitulating some of the severe disease, where what we can see is that actually, many of the animals come to infection. We can create similar atlases to the lung of these animals, and we can see that we're seeing similar things in COVID, where there's an increase in some of these intermediate progenitors, a decrease in some of the populations that we think are important for terminal function, like club cells. And so, what this suggested to us is that if we wanted to follow up on some of the hypotheses that we had, that some of these models might be appropriate. Now, there's no need to reinvent the wheel on all of this. One of the nice things that I said about genomics is that all of the data exists and it's freely accessible. One of the things that we thought was really important throughout the pandemic, and have always believed, is that everybody should be able to access the data that we generate, obviously accounting for the fact that in certain places there are ethical reasons why you would want to not share information freely. But for many of the things I'm talking about here, this is gene expression, so it's de-identified. It has no genetic information. It's just the expression of a number of genes in an individual cell. So if you want to look at any of the data sets that we generated, we have a portal called Alexandria, our library of libraries. There are other atlases that have been generated that have much of this data as well, such as the Human Cell Atlas, an organization of a number of individuals who are trying to figure out, what are the basic elements of our bodies as a basis for treating, monitoring, and diagnosing disease. And there's a special section related to COVID-19. There's also, a portal specific to all of the studies from the HCA that look at COVID-19, as well as something stood up by the Chan Zuckerberg Initiative and the Wellcome Trust. And so I put the link here for Alexandria, but all of these are readily accessible. And so if you wanted to go see where your favorite gene is expressed, what's happening in your favorite cell type, or explore other data sets, I'd encourage you to go to these portals. Many of the studies you can download the data and work with yourself. And then finally, and most importantly, I just want to thank, my team that does all this work. Obviously, as a PI during these times, my first priority in all of this stuff is sort of the safety of the individuals in my lab. It takes a lot of time to convince people that, science isn't that important, life is more important, and that, it doesn't matter if everything's delayed. And, I appreciate the individuals that want to get deeply involved in this work and, that we're keen to do it. I know for some of them, some of the people who are aspiring physician scientists, this was sort of one of the most exciting points in their life because they were sort of, able to feel like they could contribute, but at the same time, these are the great people that have kept me sane during this period and that have done a lot of this work. I want to highlight in particular, Kylie Ziegler, a very talented MD PhD student, Vince right here and Andrew, who were involved in a lot of the studies. Jose, who was one of my former postdocs and now has his own lab at Harvard Medical School, with whom we partnered on all of these studies. And so I've highlighted some of the people here that are most critical. And, I want to finish by giving our sincere thanks to all of the study participants and their families. All of sort of this understanding of what's happening biologically couldn't happen without those partnerships. And, these are things that aren't small studies. It takes, participants who are willing to contribute. It takes large collaborations among multiple different individuals at institutions that can develop technology, that can do clinical work, that can help with the bioinformatics. And I want to thank, our funders who've been incredibly generous and will hopefully continue to be. And, with that, I'll just stop and ask if there are any questions. If you're interested in some of the data sets, some of the methods, some of the technologies, I'm always happy to chat about this stuff. And would be delighted to talk about how you could apply some of this to your own research. We've done a number of different collaborations with people around the world, and would be delighted to help create capacity for you to do the things that are most interesting to you. And so with that, I'll stop sharing. I'll take any questions. Looks like I'm roughly on time. I apologize if I ran a little over.
Sure. Thank you so much for that great presentation, Dr. Shalek. We now have time for questions as well as comments. Vikas van der Merwe says, excellent work, Alex. Well done to you and your team. So that's from Vikas. And there's a question from Professor Nicola. Nikki, go ahead.
Hi, Alex. Great talk. Thank you. It was really interesting. So I missed the very beginning, but so maybe you've said this. I have two questions. One, they're both about diversity. The one being how diverse are the disease states? Because, there's so many different manifestations of COVID in terms of the disease outcome, long COVID, etc. And the other, which maybe you can't answer because you say you didn't have the genotype data, is I wonder, all these things that you're seeing with how the cells are behaving, are there known differences in some of these factors you studied across ethnic groups that might impact response to COVID?
So thanks, Nikki. And thanks, because, really, I didn't do anything. This is all my team. They're all incredible, and I'm just glad to work with them. Nikki, to your questions, when you think about long COVID, we really weren't in a position to study it here. There are many questions that we're following up on that we think are super interesting. Many people who are vaccinated are now getting infected. And so some of the ongoing studies we have are to understand what are responses sort of this initial wave of infection versus what's happening with the Delta variant, in individuals that are vaccinated or unvaccinated. So we're starting to ask that. And in other places, we're trying to get access to some tissues to figure out what might be changing in long COVID. The issue there is obviously, you want to be very careful and you have to think about sort of the ethical boundaries associated with collecting samples. One of the nice things about nasal swabs is it's easy and accessible, but many of the tissues that might be relevant if you're thinking, about some of the neural fog, et cetera, that has been associated with COVID. I mean, we wouldn't go in and obviously sample anybody's brains. So, we're trying to figure out what are the right target tissues and what we could look at there, but in some places, we'll be constrained. And, I think that this is a place where some of the animal models come in, though, whether or not some of those longer sequelae that show up in humans, are equally well represented in animals and transferable, I'm not sure yet. But, it's an area of active research. Now, the second question that you asked about the genetics, is sort of hard. This is a decent study. It's got, sort of 50, 60 participants, but it's underpowered to think about links between genetics and any responses. And, we didn't collect genotype information. There are places where people have shown that there are associations between genetics and response. For example, these places where you have mutations in some of the interferon response genes that, for example, Casanova's group is focused on, where you could imagine that because there's a defect in IRF7 or IRF3, that maybe you get a difference in ISGs. I think that more broadly, whether there are, things that would make you more susceptible or less susceptible to infection is a question that I think we'd love to ask, and I think the community would love to know. It may be that genetics is important, but I could equally easily envision that genetics is only part of the story, and what's more important is actually some of the environmental factors. And so in many places, we think a lot about how cellular composition changes as a result of diet or, lifestyle. You can imagine that high salt that shows up in some of the Western diet changes some of the composition of the gut in your epithelia. In some places, chronic exposure to allergens can change, the nasal mucosa. And so one of the big things that we're trying to do in the context of the Human Cell Atlas is to create references across different ethnic groups, across different geographies. And just to be clear, HCA is one of those things that involves people from all around the world. It's really grassroots and it's open and anybody can join. But we do have these big questions of, are there differences in baseline tissue states? Maybe it's genetics, but even more broadly, are there just changes that associate with use of particular fuels for cooking, say charcoal versus gas, that would lead to differences in susceptibility to cancers, to infections. I know that when I talk to a lot of my colleagues in Ghana or in places like Calcutta, they talk about whether or not exposure to a lot of previous infections ultimately changes the response to subsequent infection. And I think those are really tantalizing hypotheses, but they're places where we have to sort of create baseline references as a community, not only so that we can understand why particular populations may be more or less susceptible and take advantage of that to create better treatments for all of humanity, but also so that sort of these technologies and the advances that they create don't disproportionately help people in one region of the world than others. I mean, I think that it's been incredible what we can now study and what we can look at, but I'd like to see that same sort of resolution, that same sort of power accessible to everybody, so that questions and indications that are important in each region can be sort of studied in the same way, and everybody can have the opportunity to get sort of the same molecular targets. Sorry if that was a little longer than you anticipated, but I think it's a great question.
Thank you. Yeah, and your technology is amazing to be answering this question. So thank you.
Well, thank you for having me.
Thank you so much for that question, Nikki. We still have a bit of time, about like 2 minutes. So if there are any burning questions or comments, please do go ahead and raise your hand or type it in the chat box. And just to also say, we are recording this. We are going to put it up on YouTube. And it is also happening now live on Facebook. So there are some people joining us through Facebook as well. But yeah, I will send the recording and the slides, I think, if that's okay with you, Dr. Shalek, to the attendees as well. But yeah, we do have time for questions and comments as well. So please do raise your hand.
Absolutely. And if you have any questions about any of the technology, about any of the computational methods, you can just email me anytime. It's my last name, SHALEK@MIT.edu. I'm happy to chat about this stuff. I'm happy to talk about how you might adopt it to anything you might want to study. We've done a number of different diseases with partners all around the world now, and we've figured out how to make it work in a lot of places and figured out how to not make it work. And if I can save you time and money and get you started, I'd love to help in any way I can.
Sure. Thank you so much for that. I think for me, I listened to the whole presentation and I was wondering why literally you only mentioned vaccines now at the end when you were answering Nikki's question. So I was like very curious about how sort of like the cells, infected cells and the bystander cells behave differently, whether it's on a vaccinated person on unvaccinated as well? And I think you did say that you're going to be looking at that.
So it's a great question. We don't know. We're looking at it right now. We've just collected a subset of swabs that we've profiled and that we're sequencing that are coming off the sequencer to ask this question. And we have questions of what's different between sort of Delta variant versus other variants. Why do you see higher viral loads? Is it the same viral targets? Is it the same sort of impact? That's something we're curious about. We're interested in sort of what is changing in vaccinated individuals. Does infection in a vaccinated individual look different than infection in a non-vaccinated individual? And I'll just tell you from some of the studies that we did on sort of the non-human primates, there's some interesting stuff that shows up. So, we see, for example, that you might see a decrease in viral load in some of the protective therapies that show up, but actually we still see the inflammation that you would associate with untreated infection in some of the animals that are given, protection. And so, could that lead to severe disease or some of the long-term consequences that Nikki was asking about? It's possible, because of the association with the inflammation, but I think we still need to figure out, what the interventions do, how they work when they work, what's happening when they don't work. And I think that, we know that they prevent against ear disease and sort of the periphery in most instances, but we don't really know, whether or not they're giving you sort of the protection of that initial barrier that you'd want, and sort of how things are changing and what you might be able to do to actually lead to better protection moving forward.
That sounds interesting, and I'm looking forward to those findings as well. Thank you so much. I'm just going to give 2 more seconds to see if there's any more questions or comments. You can type in the chat box or raise your hand. Otherwise, I will share again, as I said, the recording and the contact details for Dr. Shalek so that you can get in touch with him if you want to collaborate or if you need any more clarity on anything that was presented on today.
Sure. So I got a private message about the computational aspects and how people might be able to do this all around the world, given sort of the fact that these require high memory and disk storage. And I think this is an incredibly important point. I will say that the computational stuff that we were showing today is actually very complicated because of sort of the size of the data. If you used to think about a study of like 50 participants being a large genomic study, you'd recognize that that's it's 50 by 30,000 matrix, and now we're talking about 100,000 by 30,000. And so, you really do need some strong computational power. And we don't actually do this on dedicated machines that we ourselves own. We've actually put all of this up into the cloud. And so, we've started using Google Cloud, which is called Terra, or there are other cloud environments like Azure from Microsoft. And so a lot of our analyses are actually done in those cloud environments. It's cheaper. You can run more machines at lower cost. You don't have to maintain the infrastructure. And so that's how we do stuff. We've actually put all of our pipelines up so you can take even some of the data sets that are showing you in the single cell portal, move them over and run your own analyses in the cloud for a limited amount of cost. But I'd be interested in hearing a little bit more about what are barriers for all of you in terms of using some of these infrastructures. I know that just because the cloud infrastructure exists doesn't mean that in every particular geography you're actually able to access it and that there aren't local constraints. But I think that one of the big questions that we've been trying to figure out in the context of the HCA and just more broadly is how do we create capacity for people to do computational work and how to do it, around some of the things that might be barriers to entry. So for us, we use cloud and we're happy to show you and share everything we have. We have a GitHub with all of our software that's freely accessible, but also happy to talk about how you might adapt some of these things to work in any particular constraints you might have so that you can get started immediately.
Okay, thank you so much for that. And I think we will definitely get in touch with you, Dr. Shalek. I'm looking at the time, and I understand that people have other meetings that they need to go to as well. And I just want to say thank you so much, Dr. Alex Shalek. I understand that in the beginning you had to do some responsibilities, and I'm grateful that you were able to join us. This was really interesting, and I'm looking forward to engaging with you further in future. And people in the comments saying, thank you, thanks for the great talk from Nikki, and other people as well in the chat. So thank you so much, appreciate it. And And to all of you, see you next month in October with a different speaker, and I'll advertise that as the time gets closer. But for now, goodbye, everybody. Enjoy the rest of your day or evening, depending on where you are in the world. Goodbye, everybody.
Thanks, everybody, for having me. Have a great one.
Thank you so much. Bye.
Bye.